Hi, my name is Linus. I graduated from CIS in 2016, and I'm now studying medicine at the Chinese University of Hong Kong, class of 2022. I recently took my IB exams and achieved a 42 in my final exams, and I'm hoping to teach biology. I also achieved an A in my EE and also a 7 in my IA, so I'm quite familiar with the IA and EE guidelines in biology. My friends also used my exam notes for the final exams, and I also helped teach them concepts before the final exams. Today I'm going to be teaching section 6.5 of the IB biology syllabus, particularly focusing on the neurons. So I'm going to be teaching section 6.5 from the IB biology syllabus, particularly points neurons pump sodium and potassium ions across their membranes to generate a resting potential. An action potential consists of depolarization and repolarization of the neuron. Nerve impulses are action potentials propagated along the axons of neurons, and propagation of nerve impulses is the result of local currents that cause each successive part of the axon to reach the threshold potential. So to start off, I'm going to explain what a neuron is. So a neuron is a excitable cell that's located within the nervous system. So it's along your spinal cord and also in your brain, and it's kind of what facilitates. It's the kind of you can think of it as kind of like a wire that helps propagate signals from the brain to effectors such as your muscles. So when a axon needs to send a signal, they'll receive what's called neurotransmitters. So these would be kind of neurotransmitters. And these neurotransmitters would bind onto channels that are located on the dendrite. And depending on what kind of neurotransmitter it is, it will fire an excitatory or inhibitory signal. But most of the time, it's going to be excitatory. So for example, if this neurotransmitter binds onto this channels located on the dendrite, the summation of these sort of signals coming from all these different dendrites will cause the axon, if it reaches a certain threshold, it will cause the axon to propagate an electrical signal down the axon. So over here within the cell body, there are actually what's called chemically gated sodium channels that would receive these signals and start to open up, and that would be the beginning of an action potential. So how an axon actually fires an electrical current is through sodium and potassium channels. These chemically gated sodium channels will begin the process of propagating action potential down the axon and towards the axon terminals, where it'll fire more neurotransmitters or either go to an effector to cause an action to be performed. The axon is also wrapped in fat, what's called a myelin sheath. So these myelin sheaths are actually produced by Schwann cells that wrap along the axon in order to speed up the propagation of action potentials. So that's kind of the first point of how neurons pump sodium and potassium. So it'll pump sodium in and potassium out to generate a resting potential and also to help propagate action potential down the axon. So in terms of what an action potential is, like the syllabus says, it consists of a depolarization and repolarization of the neuron. So what depolarization actually means is basically the neuron at rest has a negative, the inside of the neuron, the nerve axon, has a negative charge relative to the outside. So what a depolarization is when the inside becomes more positive relative to the outside, and repolarization is when the outside becomes more positive relative to the inside. So an action potential is kind of like it's this electrical signal it's a depolarization event that's propagated along the axon in order to kind of send signals down an excitable cell. So at rest, the neuron actually still has an electrical potential of -70 millivolts. And this is maintained by what's called a sodium potassium ATPase pump. And this pump pumps out 3 sodium ions in return for 2 potassium ions back into the neuron. And you would think that because it's pumping an uneven ratio of positive charges, that it would kind of upset the resting membrane potential. But because there's a high driving force for sodium to move into the cell, more so than for potassium, it will start to, the sodium will kind of slowly move back into the cell and kind of maintain the resting membrane potential. There's also what's called leaky potassium channels that would leak potassium out of the cell and into the intermembrane space. But because there are these leaky potassium channels, the potassium that's actually pumped in by the ATPase pump will start to leak out faster than sodium can move in, because sodium, although it has a higher driving force, is less permeable to the neuron. So it'll try to move in, but it'll take a lot more time for the sodium to move in. So that's how this resting membrane potential is established. So like I said earlier, an action potential is like a large depolarization event. So it's where the inside of the neuron becomes more positive relative to the outside. And it's maintained by these sodium channels. So the orange is kind of a sodium channel, and the purple is a potassium channel. Okay, so normally when the neuron is at rest, these voltage gated, because they're dependent on voltage, these voltage gated sodium channels actually remain closed. So no matter how much the sodium tries to move in, it can't move into the cell. Likewise, these potassium channels are closed. So as potassium tries to leak out, although there's these leaky channels, they can't actually leak out from the higher concentration of potassium channels to kind of offset the resting membrane potential. But when the neurotransmitter, like I said earlier, so if we go back to that diagram, when the neurotransmitter moves into the cell and kind of stimulates these chemically gated sodium channels to open. So because these chemically gated sodium channels open, some of these sodium channels will open and cause an efflux of sodium out of the it will cause sodium to move into the cell. So sodium will start to flood into the cell and make this area more positive. So it's locally more positive. And that's what establishes what's called a local current. And because there's more positive charge here than here, for instance, it creates an electrical gradient and also a chemical gradient that causes sodium to move down the axon and cause it to successively open these voltage-gated sodium channels. So, as I said, as sodium moves in, right, it'll start to make this more positive and the outside more negative. So it's starting to become more positive as it moves down, becomes more positive. And that actually causes this signal, this positive signal, causes these voltage-gated sodium channels to open. So to open. And that will cause further sodium ions to move into the axon and further propagate the action potential along the axon. So if we look at this, this graph that graphs out membrane potential to time, at the first stage, when it's at rest, it's at negative 70 millivolts. So as it's at rest, it's at negative 70 millivolts. And when these chemically gated sodium channels open because of the neurotransmitters, they'll slowly start to depolarize the axon. So the the axon will become less negative or more positive because these sodium channels are open and sodium is moving in. Once it reaches this this potential of negative 55, like I said earlier, because these sodium channels are voltage gated, they'll respond to this negative 55 millivolt threshold, what's called threshold potential, and this negative 55 will cause the voltage gated sodium channels, which are highly concentrated on the axon membrane, to open, and that will cause further sodium ions, like seen here, to flood in and cause it to become very, very positive on the inside. So that will cause a rapid depolarization. So moving on to the next section of an action potential. So as the membrane becomes more depolarized here, it will eventually reach this threshold of positive 30 millivolts. And when this threshold is reached, these sodium channels will close, and no further sodium can go into the membrane. So it's blocking further sodium from moving in. And because the inside of the neuron is more positive relative to the outside, it'll start to cause these. They'll start to cause the potassium that couldn't flow out before. Like this threshold of positive 30 actually opens these voltage gated potassium channels and cause them to move out of the cell. So these potassium ions will start to move out of the cell and cause the inside to slowly start to become more negative relative to the outside. So if we go back to the graph, it'll slowly, this is what's called the repolarization. So it will repolarize the neuron, which means to make it more negative relative to the outside, and slowly bring it back down. And eventually, because so much potassium is moving out, it'll actually do what's called an undershoot. So it'll become more negative than the resting membrane potential. And when it does this, both of the potassium and sodium channels will close. So no potassium can move out of the neuron, and no sodium can move into the neuron. And that prevents the neuron from kind of firing another action potential as one is just being propagated. So it prevents your effectors, such as your muscles, from being overstimulated. And there are actually some drugs, such as like mustard gas, that kind of cause this mechanism to kind of, it causes your neurons to become overstimulated, and it can eventually cause paralysis. So that's, the undershoot is kind of like a safety mechanism to prevent things like that from happening, overstimulation. So this undershoot causes these channels to close, and for the resting membrane potential, because if you may remember, we still have the sodium-potassium ATPase and also potassium leaky channels that will slowly reestablish the resting membrane potential. So slowly reestablish the resting membrane potential, and that's how you get back to the resting membrane potential of having the outside being more positive relative to the inside, and having a resting membrane potential of negative 70 millivolts. So the three key membrane potentials you want to remember is that rest is negative 70 millivolts. Your threshold potential that causes your sodium channels to open. So threshold for sodium is negative 55, and that causes the rapid depolarization. Then there's another threshold potential for your potassium channels to open and for your sodium channels to close of positive 30 millivolts. And that will cause it to go back to your resting membrane potential. So I hope that helped with section 6.5 of the the IB biology syllabus, and I look forward to teaching you guys.